Hi everyone, my name is Rishab Jain, and for my final project in the CSCI S-89B Introduction to Natural Language Processing class, I'll be presenting my final project, which is Natural Language Processing for Healthcare, in which I created an automatic structuring of clinical trial abstracts using sentence classification. In this project, I explored how natural language processing can be applied to medical text to automatically organize clinical trial abstracts into their standard sections. So for clinical trial abstracts, they follow a standardized structure, which include a background section, objective section, method section, results section, and a conclusion section. Despite the standardization, writing, organizing, and reviewing these abstracts is still largely manual. It's a manual process, and so these sentences can easily be misplaced, and reviewing large volumes of literature can be time-consuming. And so the goal of this project was to automatically classify individual medical sentences into their appropriate abstract sections, which hopefully reduces manual effort and improves consistency. What this project does is it uses natural language processing to analyze medical text. It takes individual sentences as input, um, predicts which abstract section the sentence belongs to, and then it can automatically structure unorganized clinical trial text. So at a high level, it basically processes and analyzes medical sentences and can predict which abstract section each sentence belongs to. Each sentence is treated as a standalone input, and the model outputs one of the 5 section labels. So this allows unstructured or semi-structured medical text to be automatically organized into a structured abstract format. So why does this matter in healthcare? Um, this task is particularly relevant in healthcare and clinical research, where documentation quality and structure are critical. Automating sentence classification can help medical writers during drafting, assist researchers during literature review, and can also support quality checks for clinical trial documentation. Especially in healthcare, any small efficiency gain can be impactful given the volume of medical literature produced each year. So my methodology for this project, I used the PubMed 20K RCT dataset, which is a publicly available dataset of randomized controlled trials abstracts from PubMed. Each sentence in the dataset has been manually labeled as background, objective, methods, results, or conclusions. And so this makes for a well-suited supervised learning and avoids privacy concerns associated with real clinical notes. The input to the model is a single sentence from a clinical trial abstract. And so, for example, I put in the primary objective of this trial was to evaluate the efficacy of metformin on glycemic control. Um, And then the output is one of 5 labels, either background, objective, methods, results, conclusions. So in this example, in output would have been objective. For the dataset size, the training set was about 180,040 sentences. The validation set was 30,212 sentences. And the test set was 30,135 sentences. And using this large data set allowed for stable learning of a deep learning model, and also there was robust evaluation. For the install and configuration, I used Google Colab, which had Python version 3.1. The GPU on it was the NVIDIA T4 with a 16 gigabyte VRAM, with a RAM of about 25 gigs. And then the operating system was Linux, which is the Google Colab default. For the model and the program, I used TensorFlow or Keras. I used NumPy. I used Pandas libraries. I used Matplotlib for graphing. I used Seaborn, and then I also used scikit-learn. For the dataset download and parsing, I got my dataset from GitHub, which has, collected data set of PubMed RCT abstracts, which was made by Frank Dernancourt. Um, and so the files were then a train.txt, a dev.txt, and a test.txt. And for the parsing, I started by separating abstracts by lines, starting with the 3 hashtags. And then each valid line had the following sequence of label and then sentence. And then I created a custom Python function to parse sentences and labels. For the pre-processing, I used tokenization using Keras tokenizer. The vocabulary in this project was limited to 20,000 words. The sentences were padded or truncated to 100 tokens, and then the labels were encoded as integers for model training. For the model architecture, I used a bidirectional long short-term memory or a BiLSTM model. And so the architecture includes an embedding layer to represent words numerically, followed by a bidirectional LSTM layer that captures context from both directions of a sentence. And this was especially important in medical texts where meaning often depends on surrounding words, which is why I use the BiLSTM model. The model then passes through dense layers and outputs probabilities across the 5 section labels using a softmax layer. For the training configuration, so the model was implemented in TensorFlow and Keras and trained on Google Colab using GPU acceleration. I used the Atom optimizer with sparse categorical cross-entropy loss, a batch size of 128, and trained for up to 10 epochs. And then early stopping based on validation loss was also used to prevent overfitting and preserve generalization. So I do have a little demonstration of the model in action. So a user can input medical text, which is first split into individual sentences. Each sentence is then passed through the trained model, which predicts its section label. And finally, the sentences are grouped into structured sentences such as methods or results. And this shows how the model can automatically organize unstructured medical text into a structured format. During the training loss decreased steadily while validation loss plateaued after the early epoch. So we can see at about 1 epoch, there's a strong increase in validation loss. And then our test performance. So on the held out test set, the model achieved a test accuracy of 81.27% with a test loss of 0.56%. The model's performance was strongest on the methods and results sections, which tend to use more standardized and technical language. And then the background and objective sentences were more challenging due to their semantic similarity. For the classification metrics, the confusion matrix highlights common misclassifications. And as you can see, most errors occurred between background and objective. Um, which often overlap in wording and intent. In contrast, the methods and results were predicted very accurately, reflecting their more distinctive structure. So, like I said earlier, we did have strong performance on methods and results, and then the more challenging classes were the background and objectives. So, what worked well? Some strengths of this approach include strong performance on technical sections, a fully reproducible pipeline, and clear practical use cases in healthcare. So the BiLSTM captured medical sentence structure very effectively. Like I said earlier, we had strong performance on the methods and results sections. And then the dataset size supported stable training, and we also did generalize quite well to the test data. What did not work so well were, like I said earlier, the background and objective sentences were often confused. And this was largely due to the overlap in language between these sections. And then sentence level classification does ignore document level context. And then there was some difficulty in classifying transitional or ambiguous sentences. Some more limitations included another limitation was that training was limited to published abstracts rather than raw clinical notes. So overall, this project demonstrated a fully reproducible NLP pipeline for classifying medical sentences into structured sections. The results show that neural models can effectively support clinical research workflows. While there are limitations, this work definitely highlights the strong potential of natural language processing in healthcare and provides a foundation for future extensions and applications. Thank you so much.